next speaker is Dr. Manjeet Rao, Professor and Deputy Director, Greenhay Children's Cancer Research Institute, University of Texas Health, San Antonio, USA. And the topic of the talk is Repurposing Antidepressant Imipramine for Treating Breast Cancer Patients.
Feel good here, then I'll go there. Let me first thank the organizing committee for giving me the opportunity to talk. Today I'm going to talk about something which we call bench to bedside. Having lost and seen the struggles of cancer patient in my own family, having lost my dad to cancer, I know firsthand what it takes basically and what exactly Dr. Tarun Krishna was talking about, an effort we all should come together to whatever we have. We can keep working on CAR T cells. We can keep working on immunotherapy, right? But at the same time, we shouldn't overlook that things are already out there to make a difference in quality of life as well as see some improvement in patient. So today I will talk about a study which was serendipitous, I would say. We were not looking for it, but that's how it happened. I'll try to finish in 20 minutes. So my lab is mostly invested in translational work. We are working on tumor microenvironment. We are working on several aspects of antibody-based therapy. But today I'm not going to talk about that. Today I'm going to talk about the repurposing of a drug. And this is a picture of San Antonio, Texas. Texas is mostly flat. And Dr. Vishwanath, you agree with me. But San Antonio is not like that. San Antonio, we call it Venice of Texas. It's a beautiful place. You guys should come and visit. If you come, please let me know. I don't really have to introduce breast cancer to this audience. It is one of the largest cancer killers in women. We know that. And we know that there are different subtypes of breast cancer depending upon the hormonal status, right? Luminal A, luminal B, HER2 positive, and triple negative breast cancer. And the prognosis in patients who metastasize is bad because of drug resistance. We don't really understand what it takes. Right? We have lots of hand waving, but we don't really understand. So these are the three issues which, if we can understand better, we can come up with some very targeted therapy, right? Metastasis, drug resistance, and not the least, toxicity to patient health, right? I have seen patients in our own clinical trials that after chemo and the nausea and the quality of life is so severely compromised that that's what we have, right? That's the chemo we have. We have been giving them for 50, 60 years, right? It doesn't discriminate between normal cells and cancer cells, right? So we have to find a better and most alternative and a less toxic treatment. So I would just introduce you that cancer cells have amazing ability to survive on an alternative way of repairing their DNA. So normal cells have very redundant mechanism. For example, if you look at triple negative breast cancer, they are deprived of some homologous recombination mediated DNA repair. There are different types of DNA repair mechanisms, right? But even though they have lost some of their arsenal, they have come up with some other solution to survive. So they use backup DNA repair pathway, right? And in that process, what they do that they incur more mutation, and they ultimately develop a clone which is not going to respond to the drug, right? So DNA repair mechanism is a fundamental process by which a normal cells become cancer, and when cancer happens, it also progresses because they have the ability to repair themselves. If you look at the mutation burden, for example, a patient given chemotherapy drug, if they have 1 in 10 mutation, after chemo, it becomes 100-fold more mutation, and no wonder that things don't work, right? So the idea was that if we can find a molecule or a protein which can work in many different DNA repair pathways. So for example, if you look at homologous recombination, there are some protein missing in triple negative breast cancer patients. But they somehow compensate that missing by increasing the expression of some other HR proteins. So if a protein involved in multiple DNA repair pathway, cancer cells most likely will be addicted to that protein, because even you remove one leg, that protein is still acting on the second leg or third leg, right? So one of such protein, and I don't really go in detail, so one of such protein is FOXM1. It is known, it was a molecule of the year in 2011 or 2013. It's a proto-oncogene because it plays an important role in many different types of DNA repair mechanisms. So we thought that if it is highly expressed in many cancers, including triple-negative breast cancer patient, ER-positive breast cancer patient, glioblastoma, you name it. It's also been known to confer resistance, for example, to cancer patients in all types of cancer patients. It has a very important role to play in HR, homologous recombination, non-homologous end joining, basal exchange repair, you name it, it plays a role. So even though cancer cells may be missing BRCA1, for example, BRCA1, the cancer cell can survive by upregulating the FOXM1 expression. So the idea was, can we target that protein? So we did some in silico screening and then followed by a compound screening. And we found a derivative of an antidepressant. We call it Imipramine blue. This is the data just showing our MST analysis and SPR analysis shows that of hundreds of compounds, this compound was the most potent hit, which looked like sitting in the pocket of FOXM1 protein because FOXM1 protein has an ability to interact among its domain for its stability. It can degrade that. We saw that it's very good degradation. FOXM1 by IV. We call it Imipramine blue. This compound is derived from an antidepressant, Imipramine, and I'll come in a minute. We have heard about depression, right, in last session. There's an important role, but nobody has really done anything systematically that we look at the patient and can we really come up with a solution for patient and does it really have any effect? So as I said, it sits in the pocket of FOXM1 DNA binding domain and it blocks its interaction between its domains, and therefore it affects the stability. So what we did basically that we did all preclinical studies, what we normally do in vivo model, and we showed that not only it blocks tumor growth, but it also blocks tumor metastasis, this compound. And we have done lots of toxicity analysis. But before I go there, we have also done ex vivo explant, where we take out patient tumor tissue from surgery directly, and without manipulating it, we grow them as if the cancer cells are growing in the body. And these cells can proliferate for 72 to 96 hours, like cancer. And then you can introduce your drug and test the efficacy of that drug in a most physiologically relevant system. Because if we had to cure cancer in mouse, we would have done it a long time ago, right? So we would want to have the most physiologically relevant tool. Ex vivo explant is one of the tools, right? Patient-derived xenograft is also good, but it's always grown in a mice which is immunocompromised, right? Immune system है नहीं उसमें, right? But with ex vivo explant, we are using in three-dimensional space. So theoretically, whatever we are seeing that it will have theoretical effect in patient, right? And when you look at basically this IB's effect, you see that we have tumor explant. You can see that in control. And when you treat with IB, the Ki67, which is a proliferation marker used for staging patient, it goes dramatically down. But if you take the normal tissue right next to the tumor tissue and then treat them with IB, you don't really see any change in the proliferation. So it's not affecting normal cells as such, right? When you look at the IB, for example, in the toxicity, in multiple tumor tissue, normal tissue, you don't really see such a problem, right? So it seems that this compound is effective, theoretically safe, right? We cannot claim it that it's safe, right? We haven't really done the more in-depth toxicity analysis before we go in clinic. So while we are working on taking this molecule in, we have just had a discussion with the FDA, pre-IND meeting, and they would like to do us to do a monkey study, and we have started this study in collaboration with NCI. While we are still doing that, it's going to take some time. What we decided basically that when I look at the structure of FOXM1 and this derivative, I thought, what about its parent compound, which is an antidepressant, imipramine? So when you model the imipramine, it's just missing this one channel, one structure here from the derivative. but it still fits perfectly in the DNA binding domain of FOXM1. Suggested us that this guy may also have the similar effect what we saw in its derivative, though it may be more potent, but it may have similar effect. And this indeed what we saw. We looked at the FOXM1 expression, and we see that FOXM1 expression goes dramatically down when you treat cells with imipramine. Then what we did preclinical study where we use the human dose equivalent of dose, which we normally use per patient with depression, which is about in outpatient setting, you can go up to 200 milligram inpatient, right? And beyond that, you have to really go in inpatient setting. And so we use different doses within 200 milligram in mouse and showed that it has a very good anti-tumor effect. It also has very good anti-metastatic effect. And of course, as it's an FDA-approved drug, it didn't have any toxicity as such, right? With a caveat, I'll talk in a minute. So then we also looked at this in ER-positive breast cancer patient, in triple-negative breast cancer patient, and we see that, yes, it decreases the proliferation, imipramine. And when we look at the triple-negative breast cancer patient and a cell line and ER-positive cell line, what we see basically there's a synergy in terms of what kind of genes are have been changed basically when you treat the cells with imipramine. And what we see that lots of DNA repair processing genes are significantly affected, suggesting that the way we had predicted, imipramine acting on the processes or proteins which support DNA repair. And if you look at the several genes basically which are downregulated in both ER-positive and triple-negative breast cancer, you see that FOXM1 target genes are predominantly affected. So that means FOXM1 is known to act as a nodal factor in, for example, in triple-negative breast cancer. 146 genes are regulated by FOXM1, sits basically in the TNBC, and that makes TNBC a TNBC. So we were excited that whatever we are hypothesizing, that like its derivative, this parent compound can also target FOXM1 and degrade it. Then the idea was that if it really affects DNA repair, let's look at DNA repair, what it happens. When we treat the cells with imipramine, we see increased DNA damage. Because cells are ultimately dying, it suggests that those DNA damage has not been repaired properly, right, as we expect. And we see that 53p foci expression levels are high. Because we know that triple-negative breast cancer patients are treated with olaparib, right, it's a PARP inhibitor. Because triple-negative breast cancer patients have high levels of PARP, and all BRCA1 mutant patient, because they are missing a leg of homologous recombination dependent DNA repair, if you hit them with PARP inhibitor, they die, cells die, right? So then the idea was, can imipramine sensitize the response? We do see very good sensitization. It's not dramatic, but it does show that there is sensitization there. And it blocks HR level, it blocks NHEG level. And then we looked at what happened, why it's working in ER-positive cell line, right? Triple-negative we can understand because DNA repair. So you may or may not know that ER-positive cancers also survive by BER mechanism. They have upregulated lots of genes in the basal excision repair pathway, right? And also alt-NHEG pathway. This is alternative back-end DNA pathway which cancer cells use. When we look at the ER-alpha signaling, we see that ER-alpha signaling is severely downregulated when cells are treated with imipramine. That means it's not only blocking the DNA repair mechanism, but it's also affecting ER-alpha signaling, which we know is a precursor for ER-positive breast cancer, right? So I'm not going to go in detail. What we're showing basically that imipramine affects FOXM1, it blocks estrogen receptor signaling, it blocks DNA repair mechanism, right? And therefore, ultimately affects the tumor growth. So based on that, we said, well, we have done the preclinical study. We have the mechanism, we know why it may be working. So we started a clinical trial. So there are 2 clinical trials currently ongoing in our center. One in breast cancer patient, another one is brain cancer patient. Because this is an antipsychotic drug, it can cross blood-brain barrier, right? So in this cohort, I just want you to focus on cohort A. This cohort here, you can see that. And now we have enrolled, 24 patients have been enrolled. And the idea was basically that we will do a window of opportunity trial where we are not changing their surgical schedule, we will just gradually increase the dose of imipramine from 50 all the way to 200, and we treat them for 28 days before they go on surgery, and then we'll harvest their tissue, look at the proliferation marker, right? So right now we have completed cohort A, and I'll show you results in a minute. We are also at the same time working on combining this with olaparib or niraparib, and also with the immune checkpoint inhibitors. I'm not going to go in detail, but it's also working in collaboration with, in combination with immune checkpoint inhibitor. So, and it was serendipitous because the result which we see, so this is the patient before the biopsy, before they were given imipramine, right? Which you can see that Ki-67 is off the roof, right? We have taken different sections, and when after the treatment of imipramine, we see a surprising decline in Ki-67 levels, suggesting that these patients are doing very good on imipramine. Here's another example of another patient. And we have seen, out of 24 patients, we have seen 17 patients have very, very good response. 7 patients had partial response, which is a pretty good idea because those patients, some of the patients were also had lung mets, and we have seen response in those patients as well. So I just want to finish my talk by first by letting clinicians here know that this drug is pretty cheap. We can make a difference here in India, right? We can start a clinical trial. If you're interested, please talk to me. We have all the IRB approvals done, and we can make a difference in patients' life without any delay. I should also mention one thing, that we had looked at 1 million women taking antidepressant in St. Louis cohort in USA, and we have seen that the incidence of breast cancer in those patients are dramatically lower, or even if they get breast cancer, the aggressiveness is a lot lower. And we know why. Because imipramine is not killing ER alpha signaling, but it's downregulating it, it's suppressing it. So in those patients where they are predisposed, for example, BRCA1 patient, right? Or for example, any other where they have patient family history, we can think about giving a prophylactic as well. We don't have to give 200 milligram, which may have some issue with heart murmur, for example, but we can give them 50 milligram or 15 milligram and put them in a path where they are not going to recur or they are not going to have tumor, right? So I encourage you, please reach out, and we can really make a difference today. And it's a pretty cheap drug. It doesn't cost anything. I would like to thank 3 main people working on this, 4 main people, Subhapriya, Santosh, Arhan. He started this work is a summer student. Is a high school student. He started this work, and that's how it led to the clinical trial. Several collaborators, Dr. Kaplamani running the clinical trial right now. Dr. Badlamudi, Dr. Jatoi is a surgical oncologist, helped us with the tumor samples. Dr. Brenner has helped me in floating our own company, and several other biostatisticians have helped us. Couldn't have done it without the support from National Cancer Institute and also the Texas Best Initiative, CPRIT. I would just leave you with one quote from one of the patients. The Melinda, she says that she's completely regressed now. I personally feel that everybody, man or woman, that's going through cancer should be on this drug, right? I'll just leave you there. Thank you.